Lecture 13, this is Module 9, and we will be discussing biotechnology. As usual, the objectives for this lecture. Biotechnology. This is the use of biological agents for technological advances. We'll be talking about a lot of different subjects in this lecture, kind of bouncing around and looking at how different aspects of biotechnology are used to help humanity in different ways. We use biotechnology widely in medicine and agriculture. We use it in other aspects as well, but these two areas definitely are big ones. DNA technology, this is a subset of biotechnology, and this is the manipulating of DNA for practical purposes. Most of what we will be discussing in this lecture will be DNA technology. So looking at using DNA, manipulating DNA to do things that we want it to do. So DNA technology, the manipulation of DNA. Scientists are able to manipulate DNA in different ways. Lots of these we will talk about. They're able to copy it. They're able to sequence it, kind of looking at the order of the nucleotides. They're able to switch it on and off. And so this is the cells can do this on their own. This is gene regulation, choosing which genes can be expressed and which ones will be not expressed. So humans, scientists have the ability to turn these on and off, as in choose which ones will be expressed or not expressed in different ways. Probe for pieces of DNA, so looking for particular pieces, certain sequences of DNA. And scientists are able to transfer pieces of DNA between species, putting one species' DNA into another. Some of these things might seem morally wrong. You might not have any issue with these. There are lots of ways that these different techniques can be used. I will not really be discussing what is the moral implications of some of these things. That's kind of for you to decide, for the scientists to decide, for the laws to decide. I'm just kind of showing you what is out there, what exists, and what we can do with things. Applications of those manipulations above of DNA technology include learning about life's history. So if we can look at how the sequences of DNA are related, if the patterns are related throughout time as organisms have gone extinct and new species have arose. Looking at learning about and looking at life's history. Relieving human suffering, so dealing with medicines and different medical procedures, protecting the environment, and enforcing the law. DNA technology can be applied to genes, particular genes of an organism, or the entire genome. A genome is all the genetic material for a specific organism. The parts of the genome or the whole thing can be manipulated. A gene is a portion of the DNA that carries information for a single protein. And we talked about this with translation, transcription, and translation. And so if you were to want to change a particular protein being made, you would focus on that particular gene. Changing those particular genes will change what proteins will be expressed. DNA is expressed in all organisms, and if you were to switch it around, potentially DNA from another organism can be expressed by all other organisms. It's basically done in the same way, depending. All organisms are doing it in the same ways. Um, in this example below, sea anemones have genes that encode for glowing proteins. A sea anemone is an animal that looks like a plant. It's found in the bottom of the ocean, in coral reef areas. Clownfish kind of live in these sea anemones. The DNA from this sea anemone was introduced into the genome of this fish. The fish is now expressing the glowing proteins. So the DNA was taken out of the sea anemone. The particular gene for this protein, this glowing protein, was taken out of the sea anemone and put into this fish. The fish is now expressing this particular protein, and you can see in the bottom right corner the fish is glowing because it's producing those proteins. The fish is now a transgenic organism. Its genome contains recombinant DNA. So the organism itself is considered transgenic because it has the DNA of another organism. The transgenic organism has recombinant DNA. Recombinant DNA is genetic material that has been spliced into another species. Transgenic organisms have the DNA of other species. Transgenic organisms have a lot of different applications, such as manufacturing pharmaceuticals. So lots of drugs that we have on the market, helping us with lots of different sicknesses, illnesses, symptoms of these illnesses lots of them come from bacteria that produce these macromolecules for us. They're massively producing these particular macromolecules that we then take, put into drugs, and sell in the market. Transgenic organisms can be used when engineering hardy crops. So crops that can withstand certain environmental pressures. You can now grow them in environments that they weren't able to grow to before. When dealing with pests, you can genetically modify them so that they are able to withstand certain pests, withstand certain pesticides, so that when you're spraying Roundup on particular crops, they can live through that where the other plants around them will not live. Transgenic organisms help when testing human genetic diseases in mice. We're able to put the genes of humans into mice so that we can use mice as the quote-unquote guinea pig for certain trials and certain experiments instead of working with humans. So how do we come about this recombinant DNA that we use? How are we able to cut the DNA at particular spots and insert it at certain spots? And so making that recombinant DNA, a lot of the times we use the help of prokaryotic enzymes. Prokaryotic enzymes are bacteria enzymes. And so a lot of the times we'll stumble across these enzymes that particular bacteria are using, and we are able to harness that and use it for our own benefits. There are lots of bacteria out there, lots of different species of bacteria, and each of those species have an array of their own proteins and enzymes. Some of those enzymes are able to protect the DNA of the bacteria by cutting out foreign DNA that might be implanted into their own genome. And so this is bacteria, like all organisms, can be invaded by different viruses. This picture here is showing a virus landing on the bacteria. The DNA is inserted from that bacteria, goes into the cell, and then finds itself, so it's inserted into the cell here, and then that DNA finds itself implanting into the genome of the bacteria. As time goes on, the bacteria will then uh, the bacteria will then replicate the DNA like it does its own DNA, but this new DNA is asking for it to be replicated over and over and over. And that's when all of these viral pieces are being made. That virus has basically taken over this cell's genome and for its own benefits. And then eventually the viruses will be made inside the cell until the cell pops open and all the viruses go spilling out to go invade another organism. So, bacteria want and have the ability to protect themselves. Some bacteria have enzymes that when they discover this DNA, they're able to cut it out. And that is where we come into play, where we take advantage of those particular enzymes. One such enzyme is called BAM-Hi. This BAM-Hi is the name of the enzyme. And so it's able to, this enzyme can cut DNA at particular sequences. Once the DNA has been cut, it leaves sticky ends. These are portions of singular stranded DNA that will be able to be attached to other sticky ends, other complementary pairs. And so if we see up at the top, this is one molecule of DNA, and then molecule B is another piece of DNA. This BAM-Hi is able to cut this particular sequence, and you can see it cuts it at a jagged end, a jagged way. This sequence is found in molecule B. This sequence is found in molecule A. It doesn't mean that these organisms, this DNA comes from the same organism. It doesn't mean that the DNA is the same specimen or closely related. This is not a very big pattern, a very big sequence, and so for it to be found all throughout the genome is very possible. And so if you were to put this BAM-Hi into DNA, anywhere that you see this sequence of DNA, this BAM-Hi would cut the DNA in a jagged way, leaving these sticky ends. Well, if you cut this molecule and you cut this molecule, then these two molecules can be put together. This sticky end and this sticky end line up complementary pairs, G to C, A to T, T to A, C to G, and they fit very well together. And then the cell itself will come in with this DNA ligase and kind of repair it and make sure that it's put together well. And so DNA with the same sequences at the same ends can be inserted into one another. So this is how we are able to cut DNA at particular sequences and put them together with other strands of DNA. Now, the length of the DNA strands can be long. It takes a lot of preparation to figure out where these cuts will be made, how they will be made, and how things can slide in. But the BAM-Hi enzyme allows for this particular cut to take place. Another way that humans can harness the making of recombinant DNA and inserting it into other organisms is called CRISPR. You might have heard this in the news talking about different experiments that are taking place with DNA. So CRISPR is a term for a Cas9 enzyme complex. Basically, this molecule is able to cut DNA at particular sequences, and then it can be used to, after it has cut DNA at particular locations, it then inserts new segments of other DNA. And so again, this complex here was discovered from bacteria. And when we found it, we then harnessed and are able to use it for our own benefit. Bacteria use it to protect themselves, but once we got a hold of it, we now can manipulate it and insert DNA into certain locations, inserting recombinant DNA of another organism into the DNA of a different organism. We are able to use plasmids to make lots of copies of recombinant DNA and proteins. So again, a plasmid is a small circular piece of DNA inside the bacteria. It is not the full genome of the bacteria, but a small piece that it has, taken in from another bacteria, or it's an extra piece of DNA, not in its nucleoid. Humans can insert recombinant DNA into that plasmid and then form a cloning vector. The bacteria will then replicate itself over and over, and the new recombinant DNA, the plasmids that are inside, will also be replicating, and every new bacteria will have this plasmid with the recombinant DNA. A lot of the times using the plasmid instead of putting the DNA into the nucleoid has all of the genes that the cell needs to do all its normal functions. If you're just putting recombinant DNA into the plasmid, you're able to not mess with the normal genome of the cell. You're just dealing with this extra DNA piece that can also be transcribed, translated, and proteins. DNA sequencing. This is a way of revealing the order of nucleotides in a gene or a genome or a piece of DNA. Sequencing DNA can be useful for deducing protein sequences and determining evolutionary relationships among species. So, if you're able to figure out what the sequence of nucleotides are, you can figure out what the protein will be if it goes through transcribing and translating. And you can also look at how closely related organisms are by the differences in their sequencing, the number of nucleotides that are different from one another, and you can calculate these differences. DNA sequencing allows us to see and show how different and similar organisms are from one another. When we look at the entire genome of an organism, let's say a human being, not all of the DNA is used to make proteins. Not all of the DNA are genes. A lot of the DNA is non-coding material. Only 1.5% of a human's genome is actually encoding for genes, encoding for proteins. That's a lot of DNA that is not used to make proteins at all. A lot of people will call it junk DNA, even though all of that is not used to make proteins, some of it are these areas of enhancers that we learned about, transcription factors with eukaryotes and things like that. But the take-home message here, only 1.5 of the human genome is actually genes that allows for the encoding of proteins. So the picture here is showing this stretch of DNA. The blue are the areas that are, the areas that will be translated to make proteins. The pink are areas that are not. And so when we look at our closest relative here, the chimpanzee, we see the sequencing in comparison to each other. And so where the blues are, they line up. So lots of proteins are the same. You've got these dips in certain spots of the non protein making areas that are different. The orangutan, you're still seeing a lot of similarities here. Then when you jump to the mouse and the zebra fish, where these proteins are, where these genes are, you still see lots of similarities. They still are lining up with one another. But where the pink is, you're seeing lots of differences. And when we get into evolution, we can kind of describe why this is, why this bit of DNA can change a lot easier than the actual protein areas will through evolutionary means. PCR is a way of replicating DNA in a test tube. So this is humans' way of replicating a small piece of DNA to make a lot of it. If you are an investigator and you only have a tiny amount of DNA and you want to make a lot more of it so that you can do lots of tests with it, this is how you could do that. Scientists will replicate DNA using polymerase chain reaction. That is what PCR stands for. So PCR will copy DNA over and over and over until you get a whole bunch of it. PCR separates the DNA strands, uses enzymes to replicate each strand, and then repeats the process over and over until it has millions of copies of the same piece of DNA in a matter of minutes, hours, a day, depending on how much is needed. Depends on how much it goes through this cycle. And so this is an example of how how PCR is done, polymerase chain reaction. So the technique, we are amplifying the DNA, so we are making copies. Amplifying is making copies. So you heat up the DNA to kind of separate them. Then you cool and add primers, just like the RNA primers that was seen in DNA replication. These primers are added to particular parts of the DNA, and then the nucleotides are added, and then you have enzymes to add these nucleotides, binding to regions and making copies. Then you have a DNA replicate. Then you do it again. You heat it up, you cool it down, add primers, add the nucleotides, and now the one piece has become 2, has become 4. The next time you do it, 8, 16, 32. In not that long, you will have millions of pieces. DNA profiling is a way of detecting differences between two individuals, either individuals of the same species or different species. And this is kind of one reason why PCR is done. So investigators may use PCR to amplify DNA from a tiny sample to a larger amount of this DNA. Once they have enough, they use DNA sequencing to detect genetic differences between individuals. So they will use PCR to make a little bit into a lot of DNA, a lot of copies. Then they'll take those copies and they'll sequence it. They'll look at the sequence of the nucleotides. Once they see the nucleotide sequence, they can then compare, making a DNA profile. They can compare different profiles to one another, seeing what's different, what is the same between these individuals. In humans and other organisms as well, there are certain areas of our DNA that have unique patterns of short tandem repeats, STRs. Um, these areas of DNA tend to be very diverse amongst individuals. So there's a lot of diversity in the short tandem repeat area compared to individual human beings. And so these short tandem repeats, just like they say, they are bits of DNA that are seen in different repeats. This individual has 1, 2, 3, 4, 5, 6, 7 repeats on one of the chromosomes. On his other chromosome, he has 9. This individual has 8 on one chromosome and 6 on the other. This one is 6 and 11. And so these are some examples showing small numbers from 6 to 11, but some of these tandem repeats can be much more. 30s, 60s, lots of different repeats. DNA sequencing shows how many short tandem repeats an individual has in their genome. So when you have sequenced DNA, or later we'll show you looking at electrophoresis, if you're able to compare these sizes to one another, you can see people's profiles. So in this person's profile, this size and this size are their profiles for these particular portions of DNA. And they are different sizes than other people's DNA. Now, when you're only looking at one, if you're only looking at one example, you're not going to find differences among every single individual. But these short tandem repeats, there are lots of different examples found within the genome. And when you compare multiple of these to 2 individuals or a few individuals, it is a lot more rare to find the exact numbers in 2 different individuals. The short tandem patterns, the short tandem repeat patterns reveal genetic differences. Comparing these STR profiles of an individual to those of a reference or to each other, you can find differences. And so these are a way of creating a person's profile. In this case, this profile is only one gene, one area of the DNA. If you are looking at lots of different particular areas of the DNA, your profiles can get much more complex. And when using this for law enforcement, that is very important. Electrophoresis is a tool that is used to separate DNA molecules based on size. So the example of the profiles that we looked at before, where the short tandem repeats were broken up, you've got someone with 6 repeats, someone with 11 repeats. These are differences in sizes of that molecule. And so when you spread them out and look at the different sizes, you can physically see that they are in different areas. There is a physical looking different profile because of the size. And we'll be using an electrophoresis in lab, and it's kind of fun how this works. So the DNA samples are put towards the negative end of the gel, and then electricity is pumped through the gel with the DNA samples. DNA is negatively charged, so it and the electricity will be moving towards the positive end. The positive end will be down here. Smaller DNA fragments will move faster than larger fragments. So when we were learning about diffusion, it's the same kind of way. It's the same kind of situation. Smaller molecules can diffuse faster than larger molecules. The example of you having a child and a large adult, and they're standing on one end of a large crowd, and they need to go through the crowd to get to the other side. The child is small, is able to push or able to weave in and out of people's legs and slip through smaller cracks and is able to get to the other side much faster than the large adult that needs to kind of push through. It takes time to move people and, excuse me. These fragments of DNA are the same way. The smaller they are able to move faster through the gel. The larger fragments move a lot slower through the gel. And so in a particular amount of time, if you give it, let's say 5 minutes, a specific amount of time where both molecules have the same amount of time to go through the gel, the smaller ones will have moved a lot further. They will be closer to the positive end, and the larger fragments will be closer to the negative end. So this is an example of the gel itself. It's kind of glowing because of the different inks that are put in here. But this is an example of a gel, and each of these lines are a different DNA sample. This is the positive end, this is the negative end, and so the pieces of DNA that are further down here towards the positive end, we know that they are smaller. The ones that are further up towards the negative, we know that they are larger. Now, to actually read a true gel is difficult to do. So a lot of the times it will be put in this kind of form that is a lot easier to kind of figure things out. And so looking at the results of an electrophoresis, the distance of the fragments that move downwards depend on the size of the fragments. So these guys down here are much smaller fragments than the ones up here. Even though, we're looking at these lines as being the same size, don't think of it as being one piece of DNA. There are lots of chunks of the same pieces of DNA after PCR is done. There are lots of copies of the same DNA, and they're all cut at the same spaces. So you'll have these lines. The molecules are the same size, but they're creating this line here. Each person has unique profile. So each of these are suspects, suspect 1 through 4. And each of these are their profiles, the differences in DNA. Each person is unique. Why? Because each person's DNA is unique from one another. And then when profiles match, that means the samples are sharing the same DNA, which mostly means they're the same person. And so this is the DNA collected at the crime scene, and this is the 4 suspects' DNA that might have done the crime. Can you see which one of these is the criminal? It is suspect 2, and we know this because everywhere the line is, this one suspect 2 shares the exact same line in all areas. Now, this suspect 2, 3, and 4, they all have this line. If this was the only piece of DNA that you were looking at from the crime, you wouldn't know who did it. But you have all of these different pieces of DNA from the scene. And so that is why you're able to pick this particular suspect out. Not all the time is this as clear cut as this example that we're seeing in front of us. A lot of the times you might have to do multiple gels over and over for a particular situation, looking at different pieces of DNA in order to know for sure. You don't want somebody to go to jail that didn't do the crime. DNA profiling has strengthened the criminal justice system over the years. Due to DNA profiling, the number of DNA exonerations in the U.S. has increased in the last couple decades. So exonerations are basically, there was DNA collected from the scene of the crime way back when, and now technology has gotten so much better that DNA can be tested against the suspect that might have been in jail for the last 2 decades. It's now discovered that DNA does not match the suspect, and that person has been in jail wrongly. So the exoneration is this idea, this concept of they're being released from jail after they were thought to be the criminal in the case. The next topic we'll talk about is cloning. And cloning is basically creating an exact copy of another organism. Now, this naturally happens in biology, in life, all the time. Cloning is basically asexual reproduction. One cell becomes two. Bacteria do it all the time. Other organisms like eukaryotes, many protistas, and plants are able to do this. Yeasts. There are even some animals that can do this. The term parthenogenesis is a term saying that an individual can either reproduce sexually or asexually, depending on what kind of environment they are in. Are there mates nearby? But cloning in this aspect, yes, it can be done naturally, asexually. But we're going to discuss how humans have harnessed the ability to clone with organisms that aren't normally able to reproduce asexually. So when it comes to human cloning, Dolly the sheep was the first animal to be cloned in 1996. It was done by this somatic cell nuclear transfer. And so we'll talk about what that means. The picture is kind of showing you are taking the egg cell out of one sheep. You are removing the nucleus from that egg cell, and we'll later learn that the nucleus from that egg cell only has half the DNA anyway, but when you take that out, it's this empty, denucleated cell. You then take a body cell, a somatic cell from another sheep. You take the nucleus out of that cell and you put it in this denucleated cell. So you have the egg, the emptied egg of one sheep, and the nucleus of the somatic cell of another sheep, and you're fusing them together. Put them under certain conditions, and you're making this egg become an embryo. The egg is normally going to become an embryo if it becomes fertilized. You're tricking the egg into thinking it has been fertilized. The cell then, starts to split, divide, and starts to become an embryo, and all of a sudden now you've got this sheep that has been cloned. The DNA is the exact same DNA as this sheep, and it is a clone of this sheep. I get it. The colors of the sheep look like this one down here. It's probably not the greatest depiction, but this is a cloned organism. Now, even though it might sound complex when I just said that, it is way more complex than that. Lots of different things needed to happen to cause this to cause Dolly to be healthy. And so adult sheep DNA was extracted and put into a different sheep's egg cell and implanted into another sheep's womb. After developing from the embryo, Dolly was born. Since then, there has been lots of other clones. Lots of animals have been cloned, repeated over and over with success. Yes, in 1996 we had Dolly, but right afterwards, in the same year, we had a mouse that was cloned. Cows, pigs, mules, all these other organisms that have been cloned. So, what are the problems with cloning? Well, the first, only a few of these clones are healthy and able to kind of live a healthy life. 3%, fewer than 3%. I've heard in other statistics, I've heard up to 10% of clones are living healthy, but that's a lot of individuals that are not living healthy. And so, with that being said, all of those individuals that something goes wrong, how much pain is that involving? Is that even, are they, I don't know. There's a lot of implications there that's kind of, how necessary is this? I'm not sure. It depends on the situation. Random mistakes can take place in gene expression, and that's kind of why some of this unhealthy death is occurring. And then there's ethical issues, ethical questions. Currently, human cloning is not legal anywhere. That doesn't mean that it hasn't been tried anywhere unknowingly. But, these questions are coming up all the time. Should scientists be able to do certain things? Why not? Things like that. This kind of leads into just the genetic engineering. And this is the altering of an organism using recombinant DNA technology. The term GMO, genetically modified organism. This is a synonym for transgenic organism, which we had learned before. Basically, a GMO, a transgenic organism, is an organism that has recombinant DNA added into its genome from another species. Or another individual. Um, an example of this is gene therapy, which we'll look at a couple slides later. Trying to cure disease by inserting a good gene to replace the mutated one. And so there are lots of good reasons to genetically modify organisms. When I say good, there are a lot of beneficial reasons, and there's also a lot of non-beneficial reasons just to see what you can do. The term genetically modified, GMO, is a buzzword, especially when thinking of food. Is the genetically modified organism able to harm the individual that's eating it? And I will touch on this a little bit because it is a question that's out there. When you eat food, you crush it up. It goes into your stomach. It gets digested. It goes into your intestines, and then those macromolecules are absorbed into your bloodstream. They go to all of your cells, and your cells are breaking those parts down and harnessing the monomers of those macromolecules to do what they need to do. Now, if there's a genetically modified organism, there's this piece of DNA that's not normally in, let's say, a strawberry. So this new gene is in the strawberry that you're eating, which ultimately means the proteins that gene is making is also in your strawberry. So you're eating a strawberry with this new protein in it. Now, chances are that the protein, I couldn't say, but hopefully that protein doesn't have any sort of toxic qualities to it. But the gene itself, just because there's an added gene into this strawberry, doesn't mean that now you're going to be able to take that gene in and it's going to go into your nucleus and it's going to be a recombinant DNA in you. You eat DNA all the time in almost every meal, and that DNA does not affect your DNA in any way. Your DNA is safe in your nucleus, and it is not going to be affected by the DNA from these things that you're eating. The proteins on the other end, on the other side, they could be. If you're, if you take the recombinant DNA of something that might be toxic, and now all of a sudden your strawberry is producing something toxic, well then yes, that's probably not a good thing. But hopefully, the agriculture or whoever is making these organisms is studying and looking at these things and making sure things are safe. But these are the questions that arise when you're buying genetically modified organisms. Um, has the due diligence taken place? Uh, GMO examples: transgenic bacteria producing drugs. So we've got the creation of insulin. So people with diabetes that need to take insulin on a daily basis or on a regular basis. That insulin is expensive to create naturally. I can't remember what organism it's taken out of, but it takes a long time for insulin to be created from a mammal. But if you make a GMO out of bacteria, that bacteria is now making all this insulin, and you're taking it out of the environment that it's living in. That is mostly how insulin is created these days. Transgenic crops, making sure that crops are able to survive the easiest with the least amount of water, the least amount of nutrition, or they can handle whatever environmental conditions they're exposed to. It makes it cheaper. It makes the groceries at the grocery store cheaper by having these genetically modified organisms. Same thing with genetically modified animals. It makes it cheaper to be able to produce more food. Is it the best for the organism's health or the organism's happiness? Those are, those moral questions that come into play. You might have heard in the news genetically modified mosquitoes. There are several different ways that mosquitoes are being modified to kind of help with the control aspects. This is just one of them. So, genetically sterile males are created in the lab, and they're released out into the wild to help control. And the reason being, we've got a normal female, a normal male. They come together and they produce healthy offspring. If you have a normal female and she mates with this sterile male, then all of the offspring will not survive to adulthood. And so adding these different genes into the genome of the males, then you are making it so any of the females that this male mates with will not be producing offspring. And so this is one way that genetically modified mosquitoes can help kind of make sure that the population of mosquitoes of this species is not going rampant. In this case, you would have to release males on a regular basis because eventually these males die, and then you have to keep up with those. There are other ways that genetically modified mosquitoes are being used, and there are a lot of people that are wary about this, and there are some genetic markers in these individuals that scientists say this genetically modified gene will not stay in the population because all of the offspring are dying. Well, there are some examples just last year, 2 years ago in Brazil. They were doing a study like this, and they found that the genetically modified gene was in 10% of a population in a certain area. And that's, how do you just how do you answer that? What do you, like that shouldn't be, but it is. And so things like that are happening. I'm not saying yes or no to this. As a mosquito control biologist, I think these different ways of controlling are incredible. There's a lot of there's a lot of hopes that these can be used in different ways. But until they're actually proven to be working well, you maybe should hold off on it. Gene therapy, we had talked about it a little bit before. This is a way of genetically modifying an organism, but genetically modifying only a few cells of the organism and not every single cell. So you take the embryo of an animal, a pig, and you change the genetics. Once that pig grows up to be an adult, every single cell in that pig has the modification. Well, gene therapy is um, focusing can focus on just particular cells of an organism. And so gene therapy is replacing faulty genes. So gene therapy may someday provide new treatment options for genetic diseases by replacing faulty genes in a person's cells. And I will come back to this word someday because someday is here. Scientists are doing this currently in different ways. Just as I'm recording this yesterday, the day before I'm recording this in 2020, they had the first human where the gene therapy was this CRISPR genetics went into a person's eye, and they're hoping that it can cut out certain portions of the DNA that are causing that person to be blind. It's a genetic disease. So things are happening slowly now, but someday is here, and they're trying these things out. So gene therapy most of the time, uses viruses. For example, cystic fibrosis is caused by a faulty gene on chromosome 7. The result is abnormal, these CFTR proteins in the lungs. Healthy versions of the gene are replaced in those cells, and the healthy genes are placed inside a virus, an empty virus that's being utilized by the doctors, and that virus goes in and it attacks or attaches to those particular cells in question, and then implants the DNA of the new protein into those cells, and then the the mutated the mutated gene gets replaced by the normal gene. Gene therapy gives cells the healthy genes. So these viruses inject the genetic material, and that genetic material is including the new healthy gene that replaces the old mutated gene. And then the proteins that gene is supposed to make is made. Cystic fibrosis in those cells is not taking place. And so this is one way that gene therapy can work.